My name is Marius. I have been at Janelia for 2 years here with the people in my lab, Karsten and Mihaly. And my lab does both a mixture of experiments and computational work. And today I'm going to tell you about one algorithm that we've recently developed called Cellpose. That's just one of the things that we work on to make the transformation from raw data that you get in your experiments to the kinds of interpretable data that we can analyze. And in particular, Cellpose arose out of a bit of a frustration with us developing segmentation algorithms for several years for calcium imaging data. But then, of course, in the lab, other kinds of data show up that we couldn't run the same tool on, so we needed something more general. And so we developed a cellular segmentation algorithm that hopefully, we think, is going to work on a very large range of types of image modalities, very different kinds of tissues and cultures and so on. And let me tell you how that works. But before I do that, I will tell you we were greatly motivated by this particular effort from 1 or 2 years ago that started in Ann Carpenter's lab at the Broad Institute, where they recognized the same kind of problem, but for nuclear images. If you look here at the bottom, you see 5 different images. They're all of nuclei, but of course they're taken with somewhat different methods. And some of them are fluorescent microscopy, others are histopathology and other kinds of microscopes. And the point they make in this this paper they wrote after this challenge, where they released lots of training data to people and had like a big Kaggle challenge to see, who can improve the most on this method. The amazing thing about it is that these, top 3 algorithms here, you can see they all improve on the reference that the challenge started from. But what's really remarkable is that all of these are one single algorithm that works on all these types of data. So in contrast, the reference is something that are pipelines, 5 different pipelines that have been optimized by image analysis experts, really, one at a time on each of these data sets. And you can see that can take a good amount of time, particularly if you're a novice at this. But now that we have these like really good models, there's no configuration time needed. They just work out of the box. So we wanted, to a great extent, to reproduce this for now segmenting cells, segmenting the cytoplasm of cells. The data set that we put together here, about 620 images or so, they are visualized here to give you a sense. These are all like little snippets from some subset of cells. They're arranged using a t-SNE plot in the middle here. The different colors are what we labeled, we thought were different kinds of image modalities. You can see they cluster to some extent according to that. And if you take a moment to look across, you'll see there's both some dual channel images, like the ones that have nuclei, so DAPI, the green and blue ones. There's a lot of grayscale. Most of these are cytoplasmic markers of one sort or another. There's some bright field microscopy in this corner. There's also somewhat that are not cells, that are actually like here you can see fruit and seashells, fruit, and so on. So we thought we'd throw a few images in there that were of the same kind of problem that we wanted to see if the algorithm would generalize to them, because we thought really what we want this generalist algorithm to do is recognize that an image is made out of many different objects, but all of the objects are very similar to each other. So that's really the underlying problem we're trying to solve. And so the practical algorithm, I'm not going to go into a lot of detail about exactly how we fit the model to do this. It's going to be fairly similar to things you've heard about before, especially here at Janelia. There's an input that's an image like this. There's an output that we generate from human labeled data. Now, these images here, these are a representation of each of the masks of the cells where the color of every pixel represents the direction in which that pixel points. And all of the pixels are trying to point towards the centers of the cell. They are effectively constructing a little dynamical system for every one of these masks, so that when we use these masks and ask every pixel to point to the next nearest pixels and next and so on, and we kind of run the dynamics of these pixels, all of those in the same mask will just go and agglomerate in the same point in the middle. And that's how we know that those pixels belong together in the same mask. All right, so that's the secret juice that made the algorithm work, is to come up with a representation of this sort that would work on cells of kind of various shapes, so that even if you're out here in these kinds of dendrites, you don't point straight towards the middle of the cell, because that would take you outside the cell. You point first towards some intermediate path that gets you to the center of the cell. And so that's the representation now that we ask a neural network to predict for us. We give it lots of training data, do lots of augmentations. We train this. And so at test time, we can just run the neural network on a new image. It gives us this representation. And then we run these pixel dynamics. We conglomerate the cells in that way. And you see here that the kind of the traces the pixels leave as they go towards the center. And then we can recover the masks very easily from that representation. And they look like that. I'm not going to tell you a lot about how the neural network is structured. It's a fairly classical standard UNet architecture. It has a few embellishments here and there to make it work as well as possible. And I'm just going to show you the results of benchmarks to try to see if we have achieved our goals. So primarily, we're going to be interested in comparing a model that I'm going to call a specialist model versus one that I'm going to call a generalist model. And the difference is that the specialist model has only been trained on this one kind of image library made of 100 images. Now, these are all very uniform. This is the data set that we actually found already before coming into this project. But these are 100 images that are fairly similar to each other, like the ones I've shown you just on the slide before. So we think that an algorithm trained just on that should be able to do very well on images from the same class, but probably will do pretty poorly on other images of cells. Whereas if we train an algorithm now on all the images of cells that we have, as well as maybe some, all these extra, non-cells but still biological images and some of the non-biological images, then we're going to see whether this generalist model does a better job of really segmenting every image you could send at it. Okay, so first, specialist model and specialist data. We also compare a few different, basically, approaches to do the segmentation problem itself. So what I have described to you, the Cellpose approach with the masks and the pixels going towards the center. So there's been at least two recent state-of-the-art approaches for doing this. One of them coming from the computer vision community. This one coming more from the image analysis community. That actually, and I'm going to come back to this. So Stefan has told us about a week ago in his talk, they've been improving on this algorithm with the authors of this algorithm that he now calls Starfinity, but we only have, for now, comparisons with this one that's called StarDist. And these are just different ways of solving the problem. So we're going to compare between frameworks, but we're particularly interested in comparing specialists and generalists. So here's the generalist models on generalized data. Now, I don't know if you can see it well enough. Here I put some arrows to places where there were some mistakes, but you can see that by and large, all these three are doing pretty well at segmenting this data, where there's lots of training data. We're not limited by training data. We're just limited by the expressive power of the model. So if a model isn't quite able to track exactly the outlines of the cells, that would be due to expressive power, not due to overfitting, because we're not overfitting there. Whereas on the bottom here, so here, this is the difficult problem. This is the one where we need to train on everything in order to be able to do well on just one new random kind of image, like the one you can see down here. And so again, Cellpose is doing very well there. This is quantified by these numbers here. There are accuracies at a threshold of 0.5. I'm going to summarize actually these accuracies right in a moment. I'll come back to the quantification of that. This is just to show you different images and the Cellpose output on them. Again, these are test images. For a lot of them, there is nothing in a training set that looks quite like it, because it's a one-off hit that we downloaded from the internet and we just kind of put into our database. And so across the board, the segmentation is pretty good. There are some mistakes here and there. We're going to come back at what to do with those mistakes towards the end. Let me quantify this for you first. If we take our specialist models, so trained only on one kind of data, and we evaluate them against specialized data, well, they're doing pretty well here. You can see at a range of matching thresholds, so this is just how well the ROI that's predicted matches the one in the training set. At a range of thresholds, all these three methods are pretty high up there in their accuracies, and we're doing a little bit better than the other ones. But then when we go to generalized data with this model, now everything just crashes. It just completely doesn't work. It just spits out gibberish because it's never seen images of that kind, so it really doesn't know what to do with them. In contrast, if we take one of these models that has been trained on generalized data, so on all these different kinds of images in our training set, then you can see two things. One, it still does as well on the specialized data as the specialist models. So we haven't lost anything. It's not like we've bombarded this model with lots of different things to do and it got worse at the things it was already doing pretty well. No, that's not the case. We're still at maximum capacity here. Whereas now in generalized data, we're starting to actually do pretty well. And you can see here, again, we are outperforming these other models, but more importantly, compare this, right, to this plot over here. So we on generalized data can now give you fairly good segmentations. Going to one of the things that I'm most excited about, if this movie starts, yeah, there we go, is we have this nice GUI that we built primarily for ourselves to be able to do the manual segmentation that Mihaly mostly did. But also now we can run the algorithm directly in the GUI, and it can give us this is the automated output of the algorithm. And we can select masks and remove them if we don't like them, and redraw them. And if we don't like everything, because everything maybe on some image, things are completely messed up. We want to start from scratch. We're going to trace these things again. And we don't need super fine detailed labels. These are pretty good. And that's the part I'm excited about, which is there's a button now to send the data to our server, where we're going to keep accumulating this training data from users and keep retraining the model iteratively. So if someone is unhappy with the quality of the segmentation because their image is slightly different, we don't quite have that in our training set, then it's very easy for them to just do the segmentation, send it to us, and then it benefits everyone when we retrain the model. And so that's the part I'm excited about. We'll see how that goes once we release this algorithm in a few weeks or so. Right now, I think we have pretty good out-of-the-box performance, no configuration time needed on many types of images. It's easy to integrate labeled images that users send us, so hopefully we can grow the training set quite a lot. We have this GUI to make it all happen, hopefully in a nicely encapsulated package. And that's pretty much the state of Cellpose. Now, that's just one of the things that we do in my lab that I'm not going to have time to get into. Mostly develop methods to process the kind of imaging data that we collect, calcium imaging data or electrophysiology data, spike sorting. So this is what you heard from Tim Harris before lunch about. And finally, another class of algorithms that I've dubbed structure discovery, which are really all about kind of taking some data, probably already processed data. You have neural activities, for example, or you have behavioral traces, and you want to like find structure in there. And these are pipelines that can do that for different data modalities.